we have the exam 3 Wednesday of week 12, which is November 13th. So that's 12 days from today. So in an effort to kind of chunk it out, what I'd like to do is my way of trying to get you to chunk it out and spend some time is let's have some very short quizzes next week, but I'm going to chunk it out. So they'll be in here like we've done some of the in-class activities. It'll just be EduCat, a couple of questions, 2 to 4 questions. And so for Monday, what I'd like you to focus on is the light reaction slide set from slide 31 to slide 46. Okay, 31 to 46. And so 31, that's just looking at getting you to think about free energy, if the sign is positive or negative. And then just the big picture idea of what's going on with redox reactions. Now, slide 38 is the full light reactions. I'm not expecting you to know that for Monday. All right. So this is showing the full thing from start to finish. We're not going to do that for Monday. That was just my way of introducing the idea of if something is in the negative portion of the redox scale or in the positive portion. All right. So don't worry about that. Basically, it's looking at redox concepts, and then we're getting into the big picture idea of what are the players in the game, what photosystems and the other big transmembrane proteins and complexes, and then how they're arranged. Okay, questions on that? So slides 31 to 46. Have a quiz on in Monday's lecture. Like I said, it's probably going to be 2 to 4 questions, but that's my way of encouraging you to chunk this out and start focusing in, because we only have 12 days. We have a lot that's going to be on that exam. Okay. And especially because a lot of what's on the exam are physiological pathways and the light reactions. It's if you haven't done some careful studying of that, it gets mixed up in your head as you take the exam. So I don't want to see that. That's very, very sad. So questions on this slide set portion? Okay. All right. I wanted to continue on with the applied uses of hormones, how do they work, and then how they've been used in real world situations. So this image is trying to get across the point that the achenes, which are all the so-called seeds on the strawberry fruit, technically those aren't just seeds, those are actually every little achene is a dry fruit. So if you've taken Plant Kingdom, you know that most of what you eat, all that lovely red sweet flesh of the strawberry is actually part of the receptacle of the flower. That's what morphs into that lovely sweet strawberry. And then the actual fruits are the achenes, and they're called dry fruits instead of fleshy fruits. Anyways, the achenes, those dry fruits, are known to be excreting out auxins, indolacetic acid, to the receptacle tissue. And that's what gets the receptacle tissue to go ahead and morph into that lovely strawberry. And so this experiment is trying to demonstrate that, where they take a normal fruit and as it's developing, what they do is, so when it's still really young, like a little baby strawberry, like what they see in figure B here, they take off all the achenes and that removes the indolacetic acid that the receptacle is getting exposed to, and you can see that it stops its development. Then if you do that experiment again, when it's a baby strawberry, you take off all the achenes, but then you spray that developing receptacle with some auxin, then it develops more like a normal fruit. So that was a nice piece of evidence to show that the achenes are excreting out and exposing the receptacle to indolacetic acid. So when they started playing around with this a little bit more and looking at what it does with flowers and fruits, they found that if you spray or expose young flowers to indolacetic acid, so some flowers are what we call perfect. That means they have both male and female structures. But if you take a young perfect flower and spray it, you essentially force it to just be female. It loses the male capabilities. So that was kind of a dramatic impact on the application of indolacetic acid. They also have discovered that indolacetic acid helps stimulate fruit set, which in unpollinated plants. So this is a big deal in industry. So if you take plants, the flowers, and they haven't been pollinated, that means technically they shouldn't be able to shift into fruit formation because you need to get pollination in order to get the sperm to the egg. And you have to get sperm and egg fusing before it'll kick into fruit development. So if you have an unpollinated flower and you spray it with indolacetic acid, you can get it to develop into fruits, but they won't have any seeds. And so we're talking about the whole production of seedless fruits, and which is a big deal. People tend to not like to have to eat fruit that has seeds in it. And so, as you know, seedless grapes, seedless oranges, the list goes on and on. The fancy word for that is called parthenocarpy. And so this is the idea that you don't get pollen to the flower, but you can still get fruit, which really is bizarre because that shouldn't happen. You should always see that you get pollination before you get fruit formation. Okay, so seedless fruit, big business. The darker side of auxins is, if you've ever heard of Agent Orange or someone in your family, parents, grandparents, they might know that term. That was used during the Vietnam War in particular to defoliate the forests. And the reason they were doing that, so they'd fly over and they'd spray Agent Orange all over the leaves. And the idea there is to get the leaves to go through abscission and fall off so they can see the enemy better and be able to detect them. And so if you have high concentrations of auxins, it does kick in leaf abscission. It's a kind of a multi-step, as we looked the other day, how it can actually kick in this case, ethylene synthesis. So it's like a toxic level of auxins. And then that stresses the plant out. They start making ethylene, which kicks in leaf abscission. You can also do this if you are a fruit grower and you want your fruit to be harvested off your plantation. Maybe it's an apple orchard. You can actually do this where you could use this as a method of pruning. So in general, you don't necessarily want all your apples on the tree to come to full maturity. You may want to take some off the tree so that the remaining fruit will develop to be bigger and sweeter. So if it looks like your particular apple tree has like too much fruit for that to happen, where you're going to end up with lots of fruit, but they'll be small and maybe they won't taste as sweet, and so it's not as marketable. So what they do then is they can use auxins as a method of pruning, and that does reduce labor costs for that. So that is an option. So they can selectively prune out some of the apples and let the rest of them get bigger and sweeter. Herbicides, again, too much of anything can be bad. And so 2,4-D, we talked about that one already as an auxin that can be used to kill plants. And what they find is that in general, a lot of monocot species of angiosperms, they can deal with that a little bit better than dicots. So they'll take that extra auxin and they'll conjugate it. And if you conjugate the hormone, that means you quiet it down. It won't be able to act. Dicot plants, on the other hand, that kind of angiosperm, the dicots, they are more sensitive to that. They can't conjugate as quickly or as much. And so if you have a lawn, for example, with your monocot grass and you want to maintain that, but some dicot weeds have popped up, spraying them with the 2,4-D or just spraying your entire lawn is okay, at least from the perspective that the monocots will survive, your dicots generally won't. And so, not that it's a great idea to spray your whole lawn with a bunch of herbicide, but a lot of people will do that. Rooting compounds, we talked about that already, that you can find that rooting hormone in stores. It can be a powder, or now they're coming out with gels or liquids that you can dip your cut stem into when you take a cutting of a plant. And then if you dip the cut portion in that and then plant it, or even just put it into some water in a glass, you'll see that the adventitious roots will start forming quite readily with that exposure to the auxin. So auxins definitely stimulate that rooting after you take a stem cutting. It's a good idea. And then the seedless fruit idea. So this is like a big list of things that we've talked about in terms of how they're using auxins in commercial applications. Okay. Cytokinins. So here we're looking at zeatin is what the plant normally makes, but they've come along with some synthetic versions that can also mimic quite readily zeatin, and they're just more stable, so they're easier to work with. We used benzylaminopurine. We call it BAP. Other people just say BA. And then there's cytokinins that have been synthetically made that are used as defoliants and herbicides. Again, we know that both auxins and cytokinins are very, very necessary in tissue culture. So whatever recipe of tissue culture that you're looking at, if you're wanting to clone a certain plant, there's going to be some form of auxin and some form of cytokinin. And it's usually specific to that plant species, what the ratio will be. But I've never seen a recipe that doesn't have those two hormones. And here's a nice picture of a tissue culture. I looked at your vessels the other day in the growth chamber, and the ones that survived past the contamination are looking quite nice. So that's nice to see. And you can see the callus formation here, all that tannish tumor looking stuff, that's your callus. And now you can actually see nice root growth and shoot growth. And that's due to stimulation with the auxins and cytokinins. So for sure, tissue culture, once they figured out what hormones they needed to use for that, that really created a big boon. It was a huge advancement in science to be able to discover how to do that in tissue culture techniques, cloning plants. That's a big deal. This is an image showing if you keep cytokinin up and running. So the bottom half are lettuce heads that are keeping the cytokinin levels high. And you can see that the heads of lettuce are greener, they look fresher. The ones here that did not get that modification, you can see they're starting to go through senescence. So cytokinin delays senescence. And you did that in lab with those spinach leaf discs that you soaked in different concentrations of BAP. And so they're finding that you can do that in real world applications. They're even finding that it can ramp up certain grain yields. And so rice, of course, is a huge grain grown for food production. And there's two major cultivars of rice that are in heavy production. One is called Japonica and the other one is Indica. And they're finding, especially with Indica, some really good results where if they manipulate it so that it has less of this enzyme, the cytokinin oxidase. So cytokinin oxidase breaks down cytokinin. So if you stop that, then you have more cytokinin. It's just more levels of cytokinin. They stay high. And they're actually finding with that manipulation, you get more seeds at the time of harvest. So here is Japonica and Indica. And you can see that you have definitely with the Indica, there's more seeds showing up. And then here, it's another picture showing the one on the right having ramped up seed production. So that, of course, more biomass, more people you can end up feeding. They've even taken this idea of, cytokinin stimulates growth. And here they're finding that they're claiming that it can be used for human uses too. So I found this at a store. I can't remember which store I was at, but I was really surprised, when it said Kinase. I'm like, wait a minute, that sounds like Kynotin. And so I picked it up and looked at it. And sure enough, they are actually putting Kynotin into their lotion. And they're claiming that it can help with skin renewal. And so especially for facial skin, they're saying that it can essentially speed up skin renewal, skin reproduction. And I don't know. I don't know if that's totally accurate or not. I'd have to look at the published studies, but a lot of that is very proprietary, so they don't really, it's not easy to find. But so they added in Kynotin, they added in resveratrol, which we'll talk about. That's a secondary metabolite that is present in fairly good doses in red wine. And that's why initially they were saying red wine is good for you because it has resveratrol, but they've backed off that stance now because now they're saying any alcohol is not good for us. They've gone back to that. And they said the amount of wine you'd have to drink to get the resveratrol's benefit is like over a liter a day. So generally shouldn't be doing that. But anyways, they are selling supplements, I guess, of resveratrol if you want your dose of resveratrol. And then sapovinol is another red wine extract. So polyphenols are found in red wine, and they're finding that polyphenols can be very potent antioxidants. So if you want to drink a liter of red wine, I would suggest the non-alcoholic versions instead, which would be way safer. So, okay. So cytokinins, really active area of research. I'll show another slide set. I'll post another slide set where they talk a little bit more about cytokinins, and they're looking more carefully at not just like skin lotion, but other ways to use cytokinin for human health. I'm not going to test you on it, but if you're curious to see how they're using plant hormones for human health, you might want to flip through those slides. Ethylene. So ethylene is a gaseous hormone. We looked at this already in the last slide set where the Yang cycle, this is an offshoot of the end portion of the Yang cycle, and it's showing the ADOMET chemical being acted on with this enzyme ACC synthase. And that will give you the intermediate product ACC. And then ACC oxidase acts on that to create ethylene. Now, ethylene we know can stimulate, can really speed up the process of senescence or abscission. And so in the cut flower industry, that is a big problem. So if you can imagine we know that ethylene is produced during wounding. When you wound a plant, whether it be a bunny rabbit munching along on the plant, or if you go and take some cuttings. As soon as you take a cutting, you cut some flowers off of the mother plant, for example, you've kicked in the wounding response. And what they're doing then is, especially in the cut area where you cut the stem, the flower stem, that's going to be a lot of release of ethylene. Well, the upshot of that is if you don't deal with that somehow, it's going to very quickly cause senescence of the flower petals, which on the right, you can see they didn't control for ethylene release. And after 14 days, you have pretty sad looking bunches of flowers. And so if you are trying to make a profit in the cut flower industry and own a flower shop, you certainly don't want to send your clients home with a situation like that, where they're just not going to come back to your store very much if they find that their bouquets senesce quickly. So that's why often, almost any time you go to a flower shop, they'll put that little powder packet with it. And you're supposed to add that into your water. And one of the reasons for that is that it will block ethylene in some manner from acting on the flowers, or at least greatly retard its ability. And so your flowers last longer, they look fresher longer, and you're happier about that. You'll go back again to that flower shop. So silver thiol sulfate will do that. It will block ethylene. And so that will slow down senescence. And so if I was looking online a while ago and they said anywhere from 15 to 35% of cut flowers are lost because of spoilage after you do the cuttings. And so, when you're a flower shop, you get the cuttings sent to you. And so already, as soon as they're cut, wherever they were initially cut, whatever business did that, and then they package them up and send them off to you. If they don't protect that process from ethylene, you're going to have definite spoilage. You're going to have some loss. So some other ideas that they have in play, depending on the business, they might do something where you can inhibit ethylene, where you essentially you have other chemicals in play that are like a mimic to ethylene, and they bind to the ethylene receptor instead of ethylene, but they don't have the same impact. So it's like a competitive inhibition. If you just have a little bit more of these kinds of chemicals that you see up top here, they can outcompete ethylene and bind into the receptor, and then ethylene can't do its job. So that's one strategy. Another strategy is if you have high carbon dioxide concentrations in the storage area where you have like the cut flowers, if you pump in a certain percentage of carbon dioxide, essentially you're trying to keep the oxygen levels low. That's essentially what you're trying to do, because if you look down here at this pathway, that last step requires oxygen. You see here that to go from ACC to ethylene, it requires oxygen. So if you can just have a room where you have the cut flowers and pump in decent amounts of CO2, you can keep oxygen levels lower and CO2 higher. Silver, we mentioned on the last slide, silver thiosulfate. So that's another strategy. Or you could do the biosynthesis. You could mess around with that. And so certain chemicals, you don't need to know the names of all these chemicals. I'm more concerned that you think about some of the mechanisms, not the actual names. But if you mess with the biosynthesis, so in this case, they found that if you block the 8-O-met to ACC, so you're messing with this ACC synthase enzyme. If you mess with that enzyme, then you can't get full completion to ethylene. Or absorption, potassium permanganate, if you add that in, that can absorb ethylene. So again, you just reduce the amount of ethylene. And I guess that's a pretty common strategy for storing apples in shipment before they make it to the grocery store. So there's several ways that they try to deal with ethylene, and it just depends what plant species, what business is working with this, that they have to deal with it. You can use ethylene though on purpose if you want to ripen up fruit at a certain time point, and you know that that fruit, that species of plant responds to ethylene and ripens up, then that's kind of nice to have that control. So maybe that way you could ship your fruit still unripe, where it's a little tougher and can handle all the bangs and bumps and dings that it's going to get in shipment. But then when it gets to its destination, you could expose it to ethylene and cause ripening right at the end, rather than before you get it on to the semi truck or the train or whatever. And so fruit ripening, if you have control on that, that would be nice. So during the fruit ripening process, when a plant is doing that naturally, it's preparing the seeds to maturity. You got to get the seeds to mature first before the plant will shift into going from an unripe fruit to a ripe fruit. So they're trying to protect those seeds. They don't want the fruit to be eaten until the seeds are good to go, fully mature, and then they're ready for dispersal. And so again, that's the way that the plant gets their seeds spread out across habitats, is if the fruit disperser comes along only after everything is matured, eats it, deposits the seeds somewhere, and then the seeds germinate and grow and hopefully establish. So you can do this with certain plants. You can expose them to ethylene, not all plants though. So this table is looking at the difference between climacteric and non-climacteric. Climacteric means they will respond to ethylene exposure and ripen up, okay? And so all of these, you can see that many of these are sold for food production. But there's a fair bit of the non-climacteric. So if you expose pineapple to ethylene, it's not going to ripen it up. Okay, so and this is something you can take advantage of. I don't know, I've lost count how many times I've gone to a store and just all you have are green bananas to pick from. The whole batch is green. And so if you want to hurry that process of ripening along, you can take an apple and cut it, because now you've wounded the apple. Ethylene is coming out, put the bananas and the apple in a bag and let them sit there for a day or two, and the bananas will respond to that ethylene and ripen up. And so that's a trick that you can do at home without too much fuss. And so there's, besides banana, there's many others on this climacteric list that you could do that with potentially. So it's a nice strategy that allows some control on the ripening process, at least for some of the plants. We already showed this, that ethylene can slow down flowering, like when the plant will actually kick into flowering, except for some key plants. It actually stimulates, it has the opposite effect in mango and pineapple, which are big production crops, but many of the others, if you time it. And so again, this is all about kind of the timing of when you want your plant to start flowering. If you have to hire people to come in and do labor for some aspect of that process, you might want to have control on when your plants are flowering. Pineapples are fun to grow. So this is a picture of, on the right is a store-bought pineapple. And this is one that PlantFit students grew a few years ago. And this is a miniature Mr. Potato Head for size reference. So you can see the pineapple didn't get very big. It's cute, but it certainly didn't meet production scale. But you can do that at home if you have space. Pineapple plants are kind of big. They're got big leaves that really arch out of their pot. So you have to have enough room. But here's what it looks like if you grow one to maturity. But you have to have some patience. So if you go to the grocery store and buy a pineapple and try to get one that has leaves that still look fairly fresh, you just cut those leaves off the pineapple fruit, and you can pot those in a pot of soil. and then just be really patient and give it some TLC and enjoy it. And it has to go through puberty essentially. So it takes a while. When we did this in the pineapple plants that we had in the greenhouse, they were grown for 2 years, I think, before we did the experiment where we put the apple slice, we cut an apple and put it into the base of the pot. Then we took a clear plastic bag and covered it all up and let that go for a little bit. And it did induce flowering. So it was a neat little trick. And unfortunately, I think because the pot was too small and it didn't have enough access to enough constant nutrients, the pineapple that was produced was small, but it was still tasty. We tasted it and it was sweet and juicy, so it was good. So that's always fun to try. But you have to have enough physical space because the plant's kind of big. Okay, so usually delays flowering, but you can actually stimulate it with pineapples. It also has a big role to play in the grain industry, so cereal grains. So that picture is just reminding you what cereal grains are. And then those are potatoes on the right. So potatoes, what you do to grow more potatoes is you don't usually start from seed, you start from the tuber. And so you take tubers, as long as they have so-called eyes on the tuber. If you plant that, it'll grow a new plant from the eyes. And so that's the buds of the tuber. Those are actually buds because the tuber is actually a modified stem. But anyway, so you can break dormancy of those and get them sprouting. It can get the seeds germinating when you expose them to ethylene. So that's another trick that you can do if you want them to go ahead and germinate and bud. So sometimes it's hard to work with ethylene because it's gaseous. And so they've come up with this liquid called Ethifon, this chemical. I think the commercial name of it is Ethrel. If you buy it, that's often how they name it. But the actual chemical is called Ethifon. And that's a liquid. It comes to you in a liquid, and you just mix that into your water that you water your plant with. And as soon as the plant takes that up in the water, it converts it to the ethylene gas. And so it's a nice way you can just add that into your irrigation, and it can allow for, you can time when you're going to do fruit ripening and fruit set. You can stimulate flowering in pineapple if you want, or delay it in other species. And so the pruning idea we talked about. So really when they are doing all of this, they're typically using the liquid, not the ethylene gas, because that can be tricky. I don't think we talked about this yet. The other thing that they noticed if you expose them to ethylene is that you tend to get shorter plants in some species. And so you can get the apical meristem slowing down, and that creates more of a compact, more just smaller plant. And then again, if you're storing food before they go out onto the grocery shelves for sale, you're trying to limit ethylene and oxygen because oxygen is needed to make ethylene. You tend to pump that room full of some concentration of CO2, the silver again, and then those chemicals that we talked about that can either absorb ethylene. You can mess with the action of ethylene by competitive inhibitors. You can stop the biosynthesis. So they've really looked at this from a lot of different angles because this is really, if you can control how much that plant sees of ethylene, that can be very beneficial to people who are in the food industry in particular. So for shipping certain fruits that are really fragile, so probably the classic example is tomato, right? If you have eaten tomatoes before, you know how fleshy they are. They're certainly not tough. You can imagine if they're bouncing around in a semi truck a couple thousand miles, of that, they're gonna get damaged, and that's a big loss. And so this idea of, well, could we ship them green? And then once they get to the destination, ripen them up. And so they have done that through, they've tried this through genetic modification. And this is the idea of antisense. Have you heard anything about antisense technology? It's basically messing with the idea of transcription and translation. So they actually use a bacteria to insert the necessary antisense gene into the tomato. And what they're doing is the antisense gene, what it does, excuse me, is it will synthesize a piece of messenger RNA that's complementary to the messenger RNA that the plant makes for ripening up. In this case, it would be ethylene, the enzymes ACC synthase or ACC oxidase. So that section of DNA that gets translated, so a piece of messenger RNA gets made. So the plant does that, right? If you introduce into that plant a complementary strand of RNA to that, it will bind to the so-called sense strand, which is what the plant made. And so you have the plant messenger RNA, and then the antisense RNA will bind to each other through complementary base pairing. Well, as you know, RNA is supposed to be single-stranded in order for the ribosomes to make sense of it. They have to be single-stranded. So if you have double-stranded RNA, it won't get made into proteins. So you've effectively stopped the process of making proteins. That's the upshot of it. And so it's a very interesting idea that works. It works really well. So basically you don't get any of that enzyme or protein made, so you won't get ethylene production. However, that tomato is still sensitive to ethylene. So you didn't mess with the receptors, but that's still fine. It's just the enzyme that you made sure wasn't going to get made so that ethylene won't get made. And so then you can ship it green, and then you can spray it with ethylene and it will ripen up once it gets to its destination. So these ideas, using antisense genes for it's usually a certain enzyme in the pathway to make that hormone. And so this was something that was probably one of the first, at least in the plant world, one of the first antisense success stories that they came up with. Okay, so ethylene, lots of ideas there. I don't know why it's doing that. I tried to erase it and it won't let me erase it. Oh, there. Okay, gibberellins. Okay, gibberellins are also big in industry. So you messed around with gibberellins in lab when you had those dwarf pea plants and the wild type pea plants, and then you sprayed them both with a liquid solution of gibberellic acid. One of the other things that we didn't have time to demonstrate in just one semester is the idea that you can speed up their puberty process, and at least some plants anyways. You can push kind of into fast forward the juvenile phase and get them into their adult portion of their life quicker, and which is a pretty big deal. They've discovered you can do that with certain conifer species, gymnosperms. So if you've ever had conifers, maybe if your parents have conifers on their property and they planted a young conifer when you were young, and if you look at it today, it's probably not a whole lot bigger compared to some other plants that can grow much quicker. So, what if you have a Christmas tree farm and you're trying to come up with lots of Christmas trees, but you have to wait and wait and wait for conifers. It takes them a while to get out of their puberty, their juvenile phase. And so this is an idea that has been attractive to people like that because you can speed that up. And the other good thing is that once you get into adult phase of life, that means you can start reproducing, which means you can start making cone tissue and get seeds in the cones. So again, if you have like, oh, this individual, I love this tree. It's perfect for a Christmas tree. It's just perfect, shape. It's beautiful. I want to get seeds of that as quick as possible so I can start planting more of it. Well, you might want to do this then, where you can get what they call precocious cone formation because you've pushed the um, timing of it that much faster from juvenile to adult. And so you get this cone formation. Here's an image of a white spruce that's still in a relatively small pot. It's quite young and it's already forming cones. That's just not natural. You would have that was done through gibberella and exposure. And then, giant sequoias, again, it takes them a long time to go into the adult phase and start forming cones. And you can see that this giant sequoia has already started as a very young seedling, very young, small, is already forming cones. And so this is something that they've known for some species anyways, you can speed up the puberty, get them into adult phase, get those cones forming, and in those cones, you'll see seeds if there's successful pollination that happens. So interesting idea. If you go into angiosperms, they found a weird thing that if you take a perfect flower again, and this time you spray them with gibberellic acid, you can force those flowers to just be male. And maybe, I don't know why you'd want to do that, but maybe that's something that's attractive for selling, or I don't know, but it's something that they've noticed. With corn plants, maize, again, it's weird because you'd think if you sprayed it young, it would just turn everything male, and instead it's just the opposite. It turns everything female. So it's just something that they noted. I don't think they use that in the real world. It's just something that they noted, which was unexpected. It also is involved in fruit set, getting apples to fully mature. They know that you're going to need to have some level of gibberellic acid. And then once that seed is mature and is hopefully lands someplace suitable, it needs to germinate to start up a new generation. And they know that gibberellic acids are part of that process. They're not the only hormone involved, but they are part of it. Okay. I think I already mentioned in the other slide set that grapevines are often treated with gibberellic acid to kind of space them out better, get them physically more room to grow bigger. And with that usually comes sweeter grapes. And so that's definitely something that people want when they go to sell their grapes. They want people to enjoy that sweetness. I guess they've even done interviews with people where they, when they have you taste something and they'll ask you to rate it, what do you think about this versus that? And so they did that with a bunch of different apples. And for some reason, it seemed that the case studies were showing that people like apples that are elongated a little bit. You might have noticed different shapes of apples being sold in stores, and some of them are longer than they are wider. And the study that they did, interviewing a lot of people on this, apparently that was a thing. And so some fruit growers do spray their apples to get them to elongate. All that it makes much difference, but some do that. And then citrus fruits, that's a big production. Lots of money goes toward that, especially in southern states. And you can actually slow down development of fruits ripening. And so it's delaying the senescence. So again, it's just a way to control the timing of it so that if you have to hire a bunch of people and come in and you have all that coordination to do for harvesting, sometimes they will do things like this. and allow. And then sometimes they stagger when they want their orchards to become mature. And so they'll have some sections, maturing up and they'll harvest that. And then they'll have another set of fields that's still young yet. And so they can use gibberellins to kind of get that to happen so that they can extend the growing season. When you're down south, you have the luxury of a longer growing season. And then that way you have more consistent citrus fruits being sent to market over a long time period. You can imagine sugarcane would be a potential good target for gibberellic acid after what you witnessed with those dwarf pea plants, how the stems really shot up and they got much taller. So of course they thought about this. Could we expose young sugarcane plants to gibberellic acid? Because when you harvest sugarcane, it's the stems that you're going for, not the leaves. You grow the plant and you're going for the stem tissue because that's where they extract the sugar from. And so we get our sugar either from sugarcane or sugar beets here in America. And if you can increase your stem length, then you should increase your sugar yield. And they definitely found that. One study I was reading, 2 tons per acre increase in the amount of sugar. So you can imagine if you're a sugar grower, that would be a good profit margin. And so getting your stems of your sugarcane plants, definitely using gibberellic acid would be a pretty easy mechanism. So we said seed germination, gibberellic acid's involved in that. Malt production, that might ring a bell with you. You might have heard about malt. And basically what we're thinking about here is that exposure of seeds to gibberellic acid kicks in alpha amylase, that enzyme. And alpha amylase is necessary to break down starch. That's its job. Alpha amylase breaks down starch. And so in the process of malt production, and in particular beer production, they might use this trick where if you've ever been on a tour, a beer making tour, they'll talk to you about the barley, grain, the hops, flour, and then of course you have to have the yeast to do the actual fermentation of all of that mixture in order to get your beer being produced. But the issue is the yeast have a hard time breaking down starch. They can do it, but it takes a long time. And so they would prefer to start breaking down glucose because that's a much simpler sugar, of course. Starch is very complex. So the trick is to get your ground malt, or barley seeds, if you grind them up, that's when they start calling it malt. And what essentially you're doing is you want to get the starch out of the malt. So you grind it up to make it easier for the yeast to attack. And then if you break down starch, it goes from starch to maltose to glucose. That's kind of the summary of the pathway. Well, then once you have plenty of glucose, the yeast can ferment like crazy because they love glucose. And they'll start fermenting that, of course, to ethanol to make the beer. I've included a couple quotes here from Benjamin, because they say that he did this quote, "Beer is living proof that God loves us and wants us to be happy." And he really didn't. That's a myth. There's a quote kind of similar to that from him, but not that one. But anyways, so you have the yeast hungry for glucose. So what they're showing here is a seed. They've dissected it where they're saying, yep, you got your baby plant inside the seed. But what you're doing is with the exposure of gibberellic acids, it gets this so-called, you don't need to know the layer here, but it's called the alluron layer. But anyways, it starts kicking up alpha amylase production. So it stimulates all of that. That's good because you break down starch. You want to get your glucose, and then the yeast can act on that. All right. And so if you ever go on a beer tour, you should ask, are you exposing the malt to gibberellic acid to speed it up? Because that yeast really will make ethanol much faster if it's got a ton of glucose versus starch. Starch just takes a lot longer to break down and ferment. And so they really want to end up with a lot of glucose that the yeast is exposed to. So this is just explaining that idea, that if you get alpha amylase, which they're showing here on the y-axis, if you get alpha amylase up and running, you can certainly speed that up if you expose that malt solution and, you know, with the yeast in there, if you expose it to gibberellic acid. You don't do that, you can see that the amount of alpha amylase is very low. And so if you can ramp up breakdown of starch, the yeast will ramp up fermentation that much quicker. So it's definitely a boon to the beer industry. It speeds everything up. So now, oh, okay, we got to stop there. Let's stop there. We'll stop with the beer production. And there's a few other things gibberellic acid has been used for, but we'll do that next week, I guess.